Hi, everyone. As you start coming in, I know there's a back up by the elevators, but I am going to begin. And don't worry, we will have everybody here before everyone gets to present. So it's just me. So I do want to thank you. The posters have been amazing. I facilitated a poster session and got to see a lot of your posters, and it's just crazy. So I apologize for all the 1,000,001 emails. This is the deadline, blah, blah, blah. But this is the wonderful things that you see. It's a lot of moving parts, a lot of really great presentations. That's pretty impressive. So I'm glad we all got here and you got to enjoy some of that. So this afternoon, this is the second portion that we're entering. But I do want to stop for a moment to recognize all of the people who have been involved with making today happen, whether it's today or your research mentors. So I get the privilege of showing you things that you may not have noticed in the larger scale. So these are the various topic areas. Oh, sorry, let me go back for a second. So the abstract book is available. I encourage you to scan this barcode, this QR code, and look at the abstract book. So these are the abstracts that the students submitted. So today we saw the posters. The abstract book will be available on the Icahn website on Monday. But if you want to get a preview, feel free to click. In the abstract book, and many of you checked off what are their topics. So I just wanted to show you the range of topics that we saw. And you're going to see all these slides have teeny tiny fonts, only because how do we fit all of this in? Because you're doing a whole range of topic. We don't restrict what areas you do research in or the kind of methodology that you do. Many of you have noticed you had first years presenting, second years, third year, scholarly year, and fourth years. So despite what some people think this is just year two, it is not. The challenge is we can't have millions of presentations because of course this would be like a weekend or a long event, but it's still pretty impressive, the breadth of the work that's been done by all of you. And so what I do want to do is thank a lot of folks who may be here. We are Zooming this, so some folks wouldn't be able to be here physically, but I do want to thank your track advisors. So these are the individuals who advised you along the way, gave you feedback, you've met with for your scholarly project. Some of them could have also been your research mentors. The MSRO team, so we have Keith and Jenny here, Grace and Yakira are probably running around. I don't see them right now, but Grace and Yakira also, we put a lot of this together. The folks who helped also with coordinating summer funding, the abstract book designers, and the volunteers today who volunteered of their time to run the elevators, make sure you got to the right room, make sure you had lunch, and so forth. Poster facilitators. So we had 20 different posters going on, 10 in session A, 10 in session B. These were various faculty that facilitated. We thank them. I'm hoping they're here, but I'm sure they're running around all over the place. And so they received, because a lot of you say, why do we have to hand in our posters so early? And what do you do with the abstracts? Well, we send them to your facilitators so they can actually be prepared, because as you know, 7 minutes and your poster, it's kind of a little hard to absorb a lot. So we try to send it to them in advance. Sometimes we have some last minute, things that come up, but in general, we try to make sure everybody is prepared. Your mentors. So I'm hoping I didn't miss anybody. And actually, I could see the very bottom isn't showing. There's one more line of individuals. So if your mentor's name is missing and it begins with after VI, I apologize. But yeah, a lot of people. So we're not saying you need to work with these 10 people. You're going out, you're meeting with folks, you're working with them, and it's just pretty amazing the breadth of the individuals. Poster presenters. So this is the second half of the day, and I wanted to take a second and just stop here. The oral presenters are amazing, but the poster presenters are amazing as well. I was very happy to sort of be able to facilitate a session and just see and hear and sort of engage in the discussion. Sometimes that's not possible, but I'm really happy. So this is all of you. Hopefully nobody's name I left off, but thank you very much. And so that's my portion at this point. This is the program for the rest of the day, rest of the time. I'm going to introduce Dr. Muller in a second. This is the lineup of the student presentations. I just wanted to highlight one thing. Sofia Ahsanuddin. Did I say it right? wrong accent, sorry. You know, I have to work hard on this. But anyway, Sofia will be the last presenter in this group with students. She's a scholar year student. The other presenters are second year students. And then Dr. Lin will be closing it out. So at this point, I would like to introduce Dr. Muller, the Dean for Medical Education. You're hesitating. Oh, okay, thank you.
Thank you, Mary. So I want to add my thanks to everyone who's contributed to this, the staff, the faculty, the mentors, and also I want to congratulate everyone who's contributed from the student perspective. I wanted to share just a little experience from today that I had just because it was timely. So for me, I mean, this is an incredibly exciting day to see all the work that you all have done and continue to do. Often in my mind, I frame it as this simple process of asking and answering questions. So today happens to be the day that I make home visits. Today I happened to be out with 2 students, and I had a couple of patients scheduled who were new, but one patient who's someone that I've known for a while. I saw her just about a month or 2 ago, and she was scheduled for an urgent visit because her home attendant called and said that one of her breasts was red. So usually it's some kind of a rash, and one of us has to look at it and figure out what it was. We went to see the patient, and she didn't have a rash. Her breast was red, and she clearly has, and she's 99 years old, born in Eastern Europe, survived the Holocaust, came here, et cetera. And for probably the last 7 or 8 years has had a slowly advancing dementia. She clearly has an inflammatory breast cancer on her left breast, which is completely new that I'd never seen before. And it was a bit of a shocker. You know, it's on your differential diagnosis, maybe, but it was a bit of a shocker. She's completely asymptomatic, so that was a relief. But it raised so many interesting questions about geriatrics and palliative care and diagnosis and treatment and prognosis. And so for me, it really brought home, in a way, what happens on a day like today, that we're confronted by things like that in medicine every day. Maybe not as dramatic as what I'm describing, but literally every day, really tough situations. And so I just want to thank all of you for asking the hard questions and for contributing to the process of answering them. So thank you very much. And I'm going to introduce Dean Charney.
So I want to add my thanks to the advisors, mentors, Mary, who does a great job, and all of you who are doing great research. I was looking at the abstracts, and as Mary showed, you cover like every area of medicine. It's incredible. And what I hope you get out of this, in terms of your research experience, not all of you are going to become researchers, but you're getting experience in asking a question and figuring out a way to solve it. And that kind of experience will help you become great physicians, even if you find out that research is not for you. On the other hand, some of you are going to find that, gee, it's fun doing research. And in some cases, you're going to do research that could help millions of people. Some of you are going to come up with new treatments for cancer, mental illness, cardiac disease. As a clinical physician, you help one patient at a time, and that's extremely rewarding, as Dr. Muller just said. And in other cases, you're going to come up with an amazing, a new research finding. And what we find from the graduates of our school, all of that happens. Some become great doctors, treating patients who need care the most, and others become great researchers. And I hope this experience is, lead you to that path wherever you find your passion. So congratulations so far. This is the beginning of your journey, and I'm sure you'll find your passion and do great things, whatever you decide. Congratulations.
So I get to introduce our speakers. Just make sure I pull this up correctly. Yep, Ashley. Let's see. So I have it up here. I do. Are they? All right, well, hopefully they'll pull it up. But I'm really pleased to welcome Ashley Brown, who is a second year presenting on her work with her mentor, Tatiana Kushner. Come on up.
Hello, everybody. My name is Ashley Brown. I'm a second-year medical student here at Mount Sinai, presenting my systematic review and meta-analysis on pregnancy outcomes in patients with Wilson's disease, both on and off treatment during pregnancy. So before I get into my project, I'd like to talk a little bit more about what Wilson's disease is for anybody who may not be familiar. So Wilson's disease, also known as hepatolenticular degeneration, is a rare autosomal recessive disorder that results in the pathologic accumulation of copper in several organs. And as we can see here, it can accumulate in the brain, the eyes, the liver, the heart, the joints, and also the kidneys, but it most notably manifests in hepatic and neuropsychiatric symptoms. And these can get so bad as to cause cirrhosis and or liver failure. So who does Wilson's disease actually affect? It's estimated that the prevalence of Wilson's disease is between 1 in 30 to 50,000 people of childbearing age in the United States as of 2020. And because it manifests in so many different ways, it can be really difficult to diagnose Wilson's disease. But once you do diagnose Wilson's disease, the treatment is to maintain their copper levels, and you can do that using copper chelating agents, like the ones that I have in parentheses here. And in really extreme cases, you may look towards liver transplant, and in those cases, that would be curative. So now talking a little bit more about Wilson's disease in pregnancy. There are existing case series and retrospective cohort studies that do look at the adverse pregnancy outcomes in patients who are diagnosed with Wilson's disease during pregnancy when you compare them to patients who do not have Wilson's disease. But there are still some gaps in this literature. There is not adequate information to really inform prenatal counseling for pregnant patients who do have Wilson's disease. And there's also very limited data on whether or not the treatment that we have for Wilson's disease actually impacts the outcomes that we're looking at. So that's why in this study, what we want to do is first determine the prevalence of key adverse pregnancy outcomes in individuals with Wilson's disease. And we also want to evaluate the impact that Wilson's disease treatment has during pregnancy on these outcomes that we're looking at in this study. So we decided to do a systematic review and meta-analysis to really pool all the literature that is available on Wilson's disease and pregnancy so that we can inform the future directions of research and so that we can better inform the prenatal care of pregnant patients with Wilson's disease. And we decided to do this using a platform called Prevalence. And for anybody who's not familiar with Prevalence, it's a great platform for systematic reviews. And it really helps to streamline the screening and really keep track of what stage you're in the process. So with input from meta-analysis expert Lily Martin in the Levi Library, we were able to query 3 different databases, Scopus, Medline, and Embase, on all literature published up until July of 2022. Our search terms included Wilson's disease or hepatolenticular degeneration, as well as pregnancy or gestation. And the screening was done by 2 voters, myself and my co-author, Sasha Kogan, in parallel, and we continue to work in parallel for the remainder of the project. We included any studies that were about pregnant patients diagnosed with Wilson's disease and reported on at least 1 outcome of interest. And all of our outcomes of interest are listed in this blue box that we have here. We excluded any studies that did not list any of the adverse outcomes of interest, any studies that were not published in English or with an adequate English translation, and also any studies that had less than 10 pregnancies included in the study. We did the full text review using the same inclusion and exclusion criteria listed on the previous slide. We did the data extraction and were able to use a form created in Excel. And then we performed the meta-analysis on that data using R with meta package. And we also use a random effects model to determine the pooled prevalence as well as a comparison between the treated and untreated groups. We also assessed the heterogeneity for all the outcomes of interest using the Q tests. We defined heterogeneity as low being 0 to 40%, moderate 30 to 40%, substantial 50 to 90%, and considerable 75 to 100%. So these are our results. We ended up with 1,482 references, which we were able to import into Prevalence, and they automatically excluded 693 duplicates. That left us with 787 studies that we were able to do title and abstract screening, and 509 were removed because they were irrelevant to our study. That left us with 277 full text studies that we were able to screen, and we excluded 258 due to the reasons that you see in the light blue box on the corner. That ultimately left us with 19 studies that we included in our systematic review. And of those 19 studies, 9 were retrospective cohort studies, 6 were case series, 4 were abstracts, and that encompassed 556 pregnant patients and 1,042 pregnancies. Then when we went to do the meta-analysis, we decided that we would leave out the abstracts. So that ultimately left us with 15 studies encompassing 429 pregnant patients and 774 pregnancies. So this table here, before we get into the individual outcomes, we do want to assess the prevalence of various adverse pregnancy and neonatal outcomes in Wilson's disease. And so we pulled the data to determine the overall rates regardless of the treatment status during pregnancy. So this data table is also organized in terms of decreasing prevalence of these outcomes. And I do want to direct our attention to the first row, where we see any adverse pregnancy or neonatal outcomes. The pooled prevalence was 33.85%. So this is basically telling us that roughly 1/3 of the patients who were diagnosed with Wilson's disease had any adverse outcome that we were looking at least one of them. And with the heterogeneity being 66%, we see that that is of substantial significance. So now before we turn to the individual outcomes of interest, this is a pooled forest plot that shows us the difference between treated and untreated patients and all the outcomes of interest. With an odds ratio of 0.64, this forest plot is telling us that patients who are treated for Wilson's disease, this is suggesting that that is a protective effect in terms of adverse pregnancy and neonatal outcomes. But we also want to keep in mind that with the p-value of 0.15, it did not quite reach significance. And we believe that may be due to the power of our study. So we do want to use this trend to see future research and if that trend continues with studies that may have more power. Now looking at individual outcomes, the first one that we saw a significant difference was C-section. With an odds ratio of 6.80, this is telling us that patients who were treated for Wilson's disease had an increased likelihood, well, it suggested an increased likelihood of having a C-section when compared to those who were not treated for Wilson's disease. And with spontaneous abortion, we see an odds ratio of 0.39. And this is telling us that patients who were treated for Wilson's disease had a decreased likelihood of spontaneous abortion when compared to those who were not treated for Wilson's disease. And with the other adverse outcomes that we were looking at, some of them did not actually cross into significance. But when we look at gestational hypertension and liver diseases of pregnancy, it's suggesting that there may be a protective factor in treating patients with Wilson's disease during pregnancy. However, when we looked at preterm birth and adverse neonatal outcomes, we did not see a difference between the two groups. So in terms of the strengths of our study, this is the first meta-analysis to evaluate Wilson's disease in pregnancy, and also to really analyze the differences in outcomes between those who were treated versus not treated for Wilson's disease during this pregnancy. This is a rigorous study design, and because we queried 3 different databases, it is comprehensive in that regard as well. In terms of our limitations, we did end up including a broad range of publication dates. Our oldest study is from 1975, and we also have 5 studies that are from the 1900s. And this is important because when we consider the demographic information that's collected in today's day and age, and we compare that to what was collected in the 1900s, there are differences in that as well. Another limitation is the study designs that we had available. There are no randomized controlled trials that are included in this study. And we also have to consider the population that we're studying, pregnant women. There are no randomized controlled trials on them right now. That left us with predominantly retrospective and case series for our study. So in conclusion, there is limited studies on Wilson's disease in pregnancy. And we believe that the heterogeneity in our study was likely impacted by the different study designs, meaning case series and retrospective cohort studies that we included, as well as the confounding variables. Also, adverse pregnancy outcomes are quite prevalent in people with Wilson's disease. As we saw in the earlier table, roughly 1/3 of patients diagnosed with Wilson's disease had some sort of adverse pregnancy or neonatal outcome. And finally, treatment of Wilson's disease during pregnancy appears to reduce the likelihood of adverse pregnancy outcomes, especially when we're looking at spontaneous abortion, but also keeping in mind the predominantly low quality evidence that we currently have. So I'm happy to say that this abstract was also accepted at DVW, and it will be presented in Chicago in May. And we also have a manuscript underway, and we do plan to update the literature search in preparation for this manuscript. I would like to acknowledge my co-authors, Marcia Lang, Sasha Kogan, and Lily Martin, and big thank you to my MSRO mentor, Dr. Tatiana Kushner, and thank you for the funding that made this project possible. Thank you so much. And at this time, I'll take any questions. Yes. Could you explain a little bit more about the Great question. So I'll repeat for anybody who didn't hear. Asking to explain more about how you diagnose Wilson's disease. Interesting enough, I was going to include that in this presentation, but because it's 10 minutes and we didn't focus on diagnosing in our presentation, I did put that in additional slides. Okay. So the AASLD released the 2022 guidelines, practice guidelines recently. And what they did was they incorporated something called a Leipzig score. Because as we were talking about earlier, Wilson's disease can manifest in so many different ways. So it's kind of hard to really just get down to the bottom of like what constitutes Wilson's disease. So using this score, they look at the ceruloplasmin levels, they look at Kaiser-Fleischer rings, which are basically when copper accumulates in your eyeball, and you can actually see that. And each of these different symptoms are assigned a number based off of this score that you can see in the corner over there. And basically you follow this flow chart, and a score of 4 or greater is a diagnosis of Wilson's disease. Great question, thank you.
Hi, thank you so much for your great presentation. Could you talk a little bit more about the genetics of Wilson's disease?
Sure. Yeah, so Wilson's disease, as we mentioned in the beginning, is a autosomal recessive condition, recessive disorder, and basically that's a mutation in a gene that's ATP-B7 gene, and that's responsible for copper transport out of the liver. And I know the second years were familiar with this, so it's a great review for STEP. Yeah, so it kind of affects the way that copper is transported out of the liver and into the bile. So copper starts to accumulate in the liver in the hepatocytes, which are the liver cells. And when they accumulate too much, they basically cause death of these hepatic cells, and that copper is then released into the bloodstream, and it can go and accumulate in other organs. And that's how you get the accumulation in the brain, producing like neuropsychiatric symptoms, in the eyes, producing the Kaiser-Fleischer rings, as well as the liver and other organs as well. Great question, thank you. Okay, yes. Okay, great question. So the question was why we ended up doing a random effects model for the pooled prevalence, and that was listed over here. The reason why we chose to do a random effects model was because we did anticipate quite a bit of heterogeneity in our studies that we included, because we were pooling together studies that were case series and also retrospective cohort studies, and also just kind of seeing the differences between those. We wanted to account for those using the random effects model. Great question, thank you. Great, thank you.
So our next speaker is Yaelle Shaked, and she's coming to talk about work that she has done with Dr. Mikey Van Gerwen. Let me pull this up for you. There you go.
Good afternoon. My name is Yasha Ked, and I'm a second-year medical student, and I'll be presenting on my summer research, Association Between Metals and Thyroid Cancer in Puerto Rico, a National Health and Nutrition Examination Survey and Ecological Study. I worked on this with my mentors, Matilda and Dr. Van Gerwen. So to start off with a background, thyroid cancer incidence rates have been increasing in Puerto Rico. According to the WHO, the age standardized incidence rate of thyroid cancer for Puerto Rico is 11.7%, whereas neighboring islands have an age standardized rate between 1.5 and 2.4%. Additionally, environmental factors are known to alter thyroid function and hormone levels. Many studies have focused on radiation and iodine exposures and new diagnostic techniques, but there are now more studies focusing on volcanic areas. And volcanic regions have been studied to have high concentrations of metals in their soils and possibly higher thyroid cancer incidence. There have been a couple studies looking at this in Italy and other places. And considering that Puerto Rico was formed by an active volcano many years ago, this got us thinking, and we soon learned about the high levels of metals in the Puerto Rican soil and the Superfund programs in Puerto Rico. So for those who don't know, the Superfund program is a program under the Environmental Protection Agency, or the EPA, and it's aimed to protect people and the environment from extremely contaminated areas. And Puerto Rico has over 20 Superfund programs throughout the island. And as you can see here, the Superfund programs are the red triangles, and they're around the San Juan metropolitan area, near Cabo Rojo, Fajardo, and kind of all throughout the island, which is a lot for a pretty small island like Puerto Rico. So this brought us to our study. We wanted to analyze the association between 4 metals: arsenic, lead, cadmium, and mercury. Sorry. And the levels of thyroid-associated antibody levels and cancer incidence in Puerto Rico. And the reason we chose these 4 metals was because these metals were found to be in high concentrations in the Puerto Rican soil in the study done by the EPA. And toxic concentration of these metals are known to alter biological processes and specifically known to be carcinogenic. For example, arsenic in high concentrations can cause microRNA dysregulation, and cadmium also in high concentrations can mimic the 17-beta estradiol on the G protein coupled estrogen receptor and promote the proliferation and invasion of thyroid cancer cells. And we chose the thyroglobulin antibodies and the antithyroid peroxidase because these antibodies have been associated with thyroid autoimmune disease. Specifically, thyroglobulin antibodies have been shown to predict thyroid cancer in those with thyroid nodules. Basically, the increased tumor cell mass in patients with thyroid cancer would release more thyroglobulin and then stimulate the immune response, which would cause an increase in thyroglobulin antibodies, showing that a positive ATG is an independent risk factor for thyroid cancer. So for the study, we used the National Health and Nutrition Examination Survey, also known as NHANES. And for those who don't know, NHANES is a large cross-sectional survey of the health and nutrition status of the US population. The survey includes a questionnaire, exam findings, lab information, and is conducted continuously, I believe, since 1999. And we used NHANES data from 2007 to 2012 and excluded patients who were less than 20 years old, participants who were taking thyroid medication, had a history of thyroid disease or cancer, or pregnant. And so our study population included around 7,695 participants. And so with this data, we created 2 models: a continuous model where the exposure to each metal was the primary predictor, and a categorical model that was divided into quartiles of exposure of the metals and adjusted for sex, age, race, smoking status, and BMI in order to control for the covariance. And then we also did an ecological study where we gathered the data on these 4 metals from a study conducted by the Environmental Protection Agency, which aimed to analyze the concentration of metals in the Puerto Rican soil and create a database of the contaminated sites. And we combined this with the age-adjusted thyroid cancer incidence rates in Puerto Rico from 2015 to 2019 from the Puerto Rico Central Cancer Registry. As seen on the right, in this map, the regions that have the darker blue shades are where there's higher age-adjusted thyroid cancer incidence rates, ranging between 29.5% and 35%, and some even have as high as 55%. So our results from the NHANES analysis showed a significant negative association between higher mercury exposure and thyroglobulin antibodies in both the continuous and the categorical models. There is no significant association between the ATG levels and the anti-TPO levels for arsenic, lead, and cadmium. And then in the ecological study, we noticed higher concentration of these 4 metals and higher age-adjusted thyroid cancer incidence rates in the northern region of Puerto Rico, as shown as in these maps. And that was also where we had the higher amounts of the Superfund programs. The Superfund programs are again shown with the red triangles. The higher levels of the metals are shown with the brown, black or dark gray circles. And then the different coloring from yellow to brown is the signaling the age-adjusted thyroid incidence rates. So in conclusion, the NHANES analysis showed a negative significant association between the levels of mercury exposure and ATG levels, which is different from what we were hypothesizing. And we believe this may be because NHANES is a cross-sectional study. And so this is one of our limitations because we're essentially looking at the metals of levels and the antibody levels in a snapshot in time. So we can't draw a causal association. And so this may have impacted our results. And additionally, in the ecological study, high thyroid cancer incidence rates and increased metal levels in the soil were observed in the northern region. And this highlights the need for more studies to investigate the role of metal exposure on thyroid health in the northern region of Puerto Rico. So thank you. Thank you all for coming. And I'd like to thank my mentors, Matilda and Dr. Van Gerwen, for helping me and supporting me in the study. Definitely wasn't easy, but we were able to put the pieces together and create the study. Thank you. Yeah.
Did you see any relationship between socioeconomic status and metal exposure and thyroid cancer in each of the regions you looked at?
Good question. That wasn't a focus of our study, but it's definitely a very interesting question. It tends to be actually that the northern region of Puerto Rico tends to be the area that has a higher socioeconomic status, just because that tends to be where the cities are located and everything. But I think that would be an excellent question to look into for future studies. Thank you.
Did you look at any other metals that happen to have high concentrations in the soil? And if so, like, were there other effects from like the presence of different metals besides the ones that you looked at?
Yeah, so from the study done by the EPA, there were other metals that were found in high concentrations in the soils, but we decided to focus on these because they're known to be carcinogenic. So we wanted to focus on these, but it would be good to look in for future studies to expand the metals that we're looking into. Yeah.
Hi, thanks so much for that. Sorry, I'm over here.
Wait,
Oh,
no.
wait.
We had a question down here.
Oh, sorry.
1 second. You'll be up next. Sorry, we're so fast.
Sorry. Did you run any correlation or was there any association in the ecological portion of the study?
So we didn't run a correlational analysis. And the reason was because we were limited on the collections, the samples that were collected and from where they're collected, and the time frame also is collected over multiple years. So kind of from method standpoint, it didn't make sense to do a correlational analysis. So that's why we kept it as a descriptive marker.
Thank you so much for that. In terms of the increase in thyroid cancer incidence rates that you saw, did you stratify that at all by staging, and level in terms of a low risk PTC versus all the way up to higher grades? And does it at all play into minerals seen here versus maybe increased screening rates and things like that?
Yeah, so that's not something that we looked into in our study. But that is, again, a great question. And I think that's something that could be included in future studies and adapted as well. Thank you for that.
Can you talk a little bit about how you came up with the idea for this study?
Yeah. So for those who don't know, I'm from Puerto Rico. And I think it's always been really important for me to bring back what I'm learning elsewhere or in school back to my home community. So I think as I was doing research and reading the literature, I noticed I was learning more about the high thyroid cancer incidence rates and thought it would be interesting to further explore this in a research study.
Great. Thank you for the presentation. I would love to know, what are the next steps with this project?
Yeah, so I think the next steps for this project would perhaps do another study looking for a causal association. But also, I think with the data that we receive from this study, it can be applied to, I think it's important for healthcare providers to kind of keep this always in the back of their mind when they're seeing individuals from these certain areas, because it is an important finding, and I think there's more research to be done in this area. Thank you.
All right. And so our next speaker, hold on, let me just get up the slides. Here we go. Sorry. All right. Our next speaker is Andrew Tieu, who is talking about work that he did with Dr. Kevin Costa. So.
There we go. Okay, hi everyone. My name is Andrew Tieu, and I'm a second-year medical student. Today, I'll be discussing some work that I did in the Costa lab over this past year, looking at the computational design of custom therapeutic cells to correct failing human cardiomyocytes. And this work could not have been done without my co-authors and mentors, Kate Phillips, Kevin Costa, and Josh Mayorian. So this presentation is adapted from some of our work that was recently published in the Frontiers in Systems Biology journal. As for some background, here's a cardiovascular disease, including heart failure, remains a leading cause of morbidity and mortality worldwide. Cell-based therapies for heart failure represent promising approaches that are actively being investigated in clinical trials. So here's a graph from the MSC heart failure trial, where heart failure patients were treated with mesenchymal stem cells. And what they saw was this modest, but still significant, increase in ejection fraction with MSC-treated patients as compared to placebo. And it's really this modest benefit that motivates a need to maximize the therapeutic efficacy of cell-based strategies. This is where computational modeling comes in. Computational modeling provides an efficient and precisely controlled method to study cellular therapies. Our lab has created a computational pipeline to investigate the heterocellular coupling effects of cell therapies in heart failure, particularly in human mesenchymal stem cells, or HMSCs, and c-KIT-positive cardiac interstitial cells, or HCICs. It has been proposed that engineering of stem cells to express specific ion channels can improve cell-based therapies. And so, using a computational modeling approach, we aim to test the hypothesis that custom hypothetical cells can be rationally designed to restore a healthy phenotype when coupled to heart failure cardiomyocytes. Moving on to our methods, we start with this computational model of the healthy cardiomyocyte. These kinds of models are great at helping us understand how perturbations and ion channels affect cardiac electrophysiology. This model in particular is the very well-established O'Hara-Rudy model of the human ventricular cardiomyocyte with all of its cardiac channels, pumps, and binding proteins. We could also make some parameter adjustments to this model in order to model a fibrotic heart failure cell. From this model, we derive a set of differential equations that we can solve using an ODE solver. And what we end up with is a simulated action potential and calcium transient. So up top is the action potential and bottom calcium transient. And in the dotted line, you can see the waveforms for a healthy cardiomyocyte. And the dotted lines represent the waveforms for a fibrotic heart failure cardiomyocyte. And note that for the fibrotic heart failure cardiomyocyte, you have a shortening of the action potential duration, as well as a smaller calcium transient amplitude. So this all so far has been for an individual cardiomyocyte. But what we're interested in is coupling it with a custom cell to see if we can improve the phenotype of the fibrotic heart failure cardiomyocyte. And so here we have our custom cell with all of our ion channels on it. And then we connect it to the failing cardiomyocyte via heterocellular coupling. And then using the power of calculus and MATLAB, we can once again generate the action potential and calcium transient for the coupled stem cell system. And then by coupling it to our custom cell, we can hopefully improve the fibrotic heart failure phenotype. What ion channels are we putting on the custom cell? Well, we put in channels from human mesenchymal stem cells, from human c-KIT positive cardiac interstitial cells, from cardiac fibroblasts, and from healthy cardiomyocytes. And then we create an initial population of 2,500 custom cell models where each of these ion channel scalings is randomly scaled from 0.01% to 1,000% of its baseline activity. And so our next question is, how do we figure out the permutations of ion channel scalings that give us the best results? We can't possibly run through every single permutation because that would just take way too long. And so instead we use a genetic algorithm, which is really just a method for solving optimization problems. And so a high level overview of this is that we start with the initial population of custom cell models that we generated with randomized ion channel scalings. And then we look at the action potential and calcium transient curves of each coupled model. From this, we can calculate the root mean squared error between the custom cell treated heart failure curve and the healthy curve. And so note here that I only show the action potential curve, but we also do this with the calcium transient as well. And over time, following a sequence of steps, we follow the parameters that minimize the root mean squared error between the healthy and the treated curves. The specific steps in the genetic algorithm include tournament, mating, mutation, elitism, and this sequence of steps is run iteratively over several generations, and there are 5 total generations being run in this case. So after the genetic algorithm, we end up with this final population of 2,500 models. And our acceptance criteria for the final models is as follows. If both the action potential and calcium transient root mean squared error were less than 50%, as compared to the root mean squared error between the healthy and untreated heart failure cardiomyocyte, the model was accepted. Otherwise, the model is rejected. And so now, diving into the results, we start with a simulation coupling fibrotic heart failure cardiomyocytes with custom cells containing channels from HMSCs, HCICs, cardiac fibroblasts, and healthy cardiomyocytes. And once again, each of these parameters for the ion channel scalings is randomly scaled from 0.01% to 1,000%. And this genetic algorithm is run over 5 generations with 2,500 models, with the initial generation being shown on the left as generation 0, and generation 5 being shown on the right. And just to orient you, on the top here, we have the action potential. On the bottom is the calcium transient. And then as before, we have a dashed line representing an uncoupled healthy cardiomyocyte and the dotted line representing an uncoupled fibrotic heart failure cardiomyocyte. As for the coupled models, we do have a number of custom cell models that are able to restore both the action potential and the calcium transient towards healthy conditions. And this is indicated by these cyan lines over here. And what this means is that by some combination of ion channel perturbations on the custom cell, we are able to improve the fibrotic heart failure phenotype. Meanwhile, the red traces represent rejected models, meaning that they were unable to restore either the action potential and or the calcium transient to within 50% of a healthy cardiomyocyte. So next, we wanted to investigate exactly which of these ion channels were most critical to accomplishing this phenotypic rescue. And so here we have the parameter fold changes for each ion channel on the accepted custom cells. For most of the parameters, the ion channel scalings span almost entire search space, so from 0.01% to 1,000%. But for these 2 parameters in particular, the accepted parameter ranges are very narrow, indicating that they may be important in rescuing healthy cardiomyocyte phenotype. These 2 parameters in particular are GCal, which represents the conductance of the L-type calcium channel, and GK1, which represents the conductance of the inward rectifier potassium channel. And so next, we ran the same genetic algorithm as before, but this time using only the 2 ion channels that we just mentioned, which once again is the L-type calcium channel and the inward rectifying potassium channel. And it's a success. Custom cells featuring only these 2 channels were able to rescue a healthy cardiac phenotype in fibrotic heart failure cardiomyocytes. This is just showing the results from generation 5, but we did run the full genetic algorithm here as before. And so now we took it one step further and we tested custom cells with only the inward rectifier potassium channel and only the L-type calcium channel. And once again, we were able to generate viable models showing restoration of both the action potential and the calcium transient, with the Icahn models in particular showing the greatest improvement. And now finally, given that custom cells expressing IK1 showed promise in correcting the fibrotic heart failure phenotype, we wanted to investigate the translational potential of our findings by testing the effects of IK1 as expressed on HCICs. So first, we couple the failing cardiomyocytes with standard HCICs, and we see that this is not sufficient to restore healthy cardiac phenotype, showing only a modest improvement in both the action potential and calcium transient kinetics. The best performing model in each of these cases is highlighted with this magenta line with the corresponding root mean squared error shown on top of it. And once again, this is just the results from generation 5, but the full genetic algorithm is being run here. However, when we express IK1 on these HCICs and then once again couple them to the fibrotic heart failure cardiomyocytes, we see a large improvement in the action potential and the calcium transient. While it doesn't quite satisfy our acceptance criteria, we see a large drop in the root mean squared error from 0.87 to 0.54 in the action potential, and from 0.77 to 0.38 in the calcium transient. And so in conclusion, computational modeling provides an efficient and precisely controlled method to study cell-based therapies. Custom cells expressing cardiac ion channels, especially IK1, are able to restore near healthy action potential and calcium handling phenotypes in fibrotic heart failure cardiomyocytes. And why is all of this important? Well, it means that maybe one day we will actually be able to engineer these cellular therapies as a treatment for heart failure. As for our limitations, we did not model paracrine signaling effects of cell therapy, and we focused on single cell action potential and calcium transient. This leads us to our future directions, which include incorporating paracrine signaling effects in the model, tissue level modeling simulations, and experimental validation studies of proposed therapeutic cells. And that's all I have. Thank you so much for listening. And I'm happy to take any questions.
Hey, okay. I was wondering if there's a way to model the conduction system in addition to the cardiomyocytes.
So the conduction system that we're modeling here is based on the gap junctions in between the stem, like our custom cell and the cardiomyocyte. And so if we were to alter this in vitro, say, this would be accomplished by altering the amount of connexin proteins that are expressed in between these two cells. And while stem cells in general have been shown to have variable amounts of connectivity with the cardiomyocyte system, this is an active area of research, and altering these kinds of connection proteins in order to improve electrical coupling in between this heterocellular coupling system.
I'm wondering if you're looking towards maybe using cell culture models to kind of validate some of these computational models and something that's a little bit more, closer to the human body.
Yeah, so I think the idea of having a custom cell is very out there, in that the reason I use the word custom cell is because there really is no biological basis to the custom cell that I'm designing, right? And that is why, in order to investigate more of a translational side of things, that's when we did the experiment at the end, putting IK1 specifically onto HCIC cells. And this is something that has been researched a little bit more in current literature. So current attempts, for example, at creating these kinds of chimeric cells include cardiochimeras. And so there's been recent papers showing that combinations and fusions of HCICs and HMSC cells injected intramyocardially for stem cell transplants have been more effective than either alone. Other examples include incorporating IK1, connexin proteins, and sodium channels into current fibroblasts. And when these are injected into a cardiac system of interstitial fibrosis, it's been shown to rescue twitch force and conduction velocity. And so this would be an example of a more grounded approach rather than a more abstract computational system.
Yeah, so you said that you weren't able to model the paracrine signaling. If you could, how would you model the extracellular signaling environment, or would that introduce a level of complexity that's unfeasible?
Yeah, so paracrine signaling has actually been modeled in certain stem cell types. The reason we didn't do it here is because in a custom cell, we would really have to determine how paracrine signaling works empirically, and we really just don't have that kind of information for an abstract cell. As for paracrine signaling in HMSCs, for example, paracrine signaling is very important and it has like very well-established effects on channels such as the L-type calcium channel, SERCA, and it's also important in anti-fibrotic effects. And actually for stem cell therapies in general in the heart, paracrine signaling and the secretome is extremely important in conducting its therapeutic effects.
Yep. Hey, Andrew. So can you tell us a bit more about what the difference is between coupling a custom cell that expresses a potassium channel to a fibrotic myocyte and administering a drug that might kind of allosterically do something to the potassium channel on the fibrotic myocyte itself?
Yeah, so I think that's kind of an abstract question. So I think something that we had originally looked at was like actually performing gene therapies in order to modulate ion channel expression on the cardiomyocyte, which I think is kind of what your question is getting at. And so I didn't show these slides, but some of our results show that you would actually have to do there's no single gene therapy that would allow you to modulate one single ion channel and get the electrical results that we want. It would be like a very complex therapy that would result like knockup and knockdown of multiple ion channels. And so that's kind of what led us to our current experiments where, okay, maybe instead of having like this very complex gene therapy protocol, we can just inject these one types of cells with these certain modulations on it. And so while the way that electrical activity is affected by these two different protocols is similar, it isn't exactly one-to-one, and we had better results with this heterocellular coupling rather than direct gene modulation.
Wonderful. And for our last speaker, we have Sofia Ahsanuddin, who's going to be speaking up about work that she's done with Dr. Rosen. Hold on, let me pull that up for her. Sophia, do you have it on your flash? It's on here. There we go. Come on.
Good afternoon, everyone. So today the topic of my presentation will be on flavoprotein fluorescence and its use as an indicator of mitochondrial dysfunction and the retinas of sickle cell disease subjects at steady state. And these are my disclosures, and these are my funding sources. So before I begin my presentation, I wanted to give you all some take-home points to consider as I go through my talk. At the end of this presentation, I would hope that you all remember that oxidative stress is essential to the pathophysiology of sickle cell disease. Vision researchers like myself can quantify mitochondrial dysfunction that can lead to oxidative stress in the human retina by means of measuring something we called mitochondrial flavoprotein fluorescence, or FPF for short. FPF is present in sickle cell patients even before there are telltale structural signs of sickle cell retinopathy that can be detected by standard retinal imaging modalities like spectral domain optical coherence tomography, or SDOCT for short. So our story begins with mitochondria, which, as we all know from our classes, are the primary energy-producing organelles of human cells. Mitochondria generate more than 90% of energy in the form of ATP, and they also coordinate numerous components of cellular metabolism, including apoptosis and calcium signaling. When mitochondria don't work properly, this can lead to the pathogenesis of various retinal disease states, such as age-related macular degeneration and diabetic retinopathy. Now, a major cause of mitochondrial dysfunction is oxidative stress, which is characterized by the overproduction of reactive oxygen species and impairment of antioxidant defense systems. Impaired cell respiration and activation of cytochrome C can ultimately lead to diminished production of ATP, and this can ultimately result in cell death or apoptosis. And it's this impairment of the electron transport chain that exacerbates baseline electron leak across the mitochondrial membrane, which further increases reactive oxygen species production from complexes 1 and 3, as shown here on the slide. So why is all of this important? Well, in human subjects, we know that mitochondrial dysfunction actually precedes changes in visual acuity and structural changes that can be detected by standard retinal imaging modalities. And so we can assess the presence of a mitochondrial dysfunction in the retina by quantifying the presence of oxidized mitochondrial flavoproteins, which, as we know, are enzyme proteins that participate in redox reactions that are fundamental to cellular respiration. The key point here is that flavoproteins become oxidized, and then this can lead to mitochondrial stress very early on in the disease process. And so earlier detection of mitochondrial dysfunction in the retina can possibly result in increased positive visual outcomes for our patients, because then we can introduce treatment earlier on rather than later on. And so in this study, we used a novel imaging device known as the Occupant Beacon to quantify the presence of oxidized flavoproteins in the human retina of sickle cell disease subjects compared to healthy controls. And so this device basically allows us to measure flavoprotein fluorescence, which, as I mentioned earlier, is a readily detectable marker of oxidative stress. The mechanism by which this device quantifies FPF is beyond the scope of this presentation, but if anyone has any questions about that, we can certainly discuss after my talk. So FPF has been previously shown to increase in a variety of retinal pathologies, ranging from diabetic retinopathy to central serous retinopathy to idiopathic intracranial hypertension. But there have been no studies conducted to date on hemoglobinopathies such as sickle cell disease, which, as we know from our classes, affects multiple organ systems throughout the body, not just the blood vessels. And so the objective of this study was to evaluate FPF in the retinas of sickle cell disease subjects. As the most common inherited hemoglobinopathy in the world, sickle cell results in dramatic alteration and function of red blood cells. We know that sickled red blood cells can obstruct the vasculature, they can activate leukocytes, they can activate platelets and endothelial cells, and they can exacerbate chronic inflammation. And so in this regard, sickle cell is not only characterized by hemolytic anemia, but it's also characterized by vaso-occlusive episodes, which are incredibly painful for these patients, and progressive organ damage. Now, in the ophthalmological community, the mechanisms that drive unrestrained inflammation and organ damage are of immense clinical interest, because traditionally, the way that we have looked at sickle cell retinopathy was that we looked at the periphery of the retina here. But in recent advances in retinal imaging modalities have allowed us to detect even earlier structural changes that occur in the macula of the retina, which is the region of the retina that is most important for central vision. And so up here on the slide here, we have two images of how sickle cell affects the human retina. On the left here, we have a fundus fluorescein angiography and a cross-section of an SD-OCT image of a patient's right eye, which revealed loss of the inner retinal, the inner retinal layers of the temporal macula, as indicated here. And then on the right here, we have an infrared image and a cross-sectional SD-OCT image of another sickle cell disease patient's right eye, which reveals thinning of the temporal outer macular area. And these are some additional images of two patients who have the HBSS genotype, which, as we know, is the more severe genotype of sickle cell disease. Up on the top here, on the upper left, we have the white arrows indicating areas of neovascularization. And on the bottom left here, we have a retinal thickness heat map showing retinal thinning highlighted in blue, and the corresponding OCT cross-sectional image of the patient's retina showing retinal thinning in the temporal macula. And so to really drive home the role of ischemia in propagating retinal thinning and capillary dropout in sickle cell disease patients, I've included here a side-by-side comparison of an ultra-wide field OCTA image of my left eye compared to the left eye of a 32-year-old sickle cell disease patient I imaged earlier this month with the HBSS disease genotype. And as we can see here on the right, we have massive amounts of ischemia and capillary dropout. that not just only affects the temporal retina, but also the macula of this patient. And so unfortunately, this patient presented with progressive vision loss and floaters in his left eye. So to quickly transition to my study, I was able to image 43 sickle cell disease patients and 46 healthy controls. And what we're able to see basically is that in sickle cell patients, we have significant elevations of flavoprotein fluorescence in the retinas of these patients compared to healthy controls. And when we stratify the sickle cell patients by disease genotype with the blue box plots showing the more severe genotypes, SS and S beta 0 thalassemia, and the hot pink box plots showing that a more mild to moderate genotypes, SC and S beta plus thalassemia, we still see a statistically significant elevation of FPF at the macula in both types of sickle cell patients. Similarly, we looked at FPF at the optic nerve head in all 43 sickle cell patients, and what we see is a statistically significant elevation in FPF in sickle cell patients compared to healthy controls. And when we stratify by genotype, we see the same elevation in FPF, regardless of how severe the manifestation of the disease is. So I had mentioned earlier that many sickle cell patients present with thinning in the temporal retina. So we evaluated whether we can detect changes in FPF even before retinal thinning can be detected on standard retinal imaging modalities like OCT. And so what's fascinating to me about this study is that despite comparable retinal thickness in the nasal outer subfield and the inferior outer subfield, as shown in the white circles on the left graph, we can still appreciate significant elevations in FPF in those same ETDRS subfields of the macula. And so I think this pretty concretely demonstrates the presence of mitochondrial dysfunction even prior to any structural alterations in the retinal architecture. And so these are a couple of tables demonstrating the retinal thickness indices of sickle cell patients compared to healthy controls. What we can see here is that sickle cell patients have comparable retinal thickness compared to controls in the temporal outer macula, the nasal outer macula, the inferior outer macula, and the central macula. But when we look at FPF, we see significant elevations of FPF in those same ETDRS subfields of the macula. So because we know that retinal ischemia can lead to increased levels of oxidative stress and subsequent vision loss in sickle cell patients, we also sought to evaluate the correlation between FPF and various capillary perfusion parameters as measured by OCTA. And so again, the way that we were able to quantify intermittent perfusion of the retinal microvasculature is beyond the scope of this presentation. But what we were able to do was we were able to quantify something we call intermittent perfusion of the capillaries in the macula of these patients, with red indicating capillary non-perfusion and cyan indicating capillary reperfusion. And so what we find here is rather interesting. We find that as FPF increases, capillary density decreases, which makes sense. We know that as there are increased levels of oxidative stress, we see increased capillary dropout in these patients in the same region of the macula. Similarly, as FPF increases, we see an increase in intermittent perfusion of the retinal microvasculature. And again, FPF is significantly correlated with capillary reperfusion in the macula, as well as non-perfusion in the macula. So lastly, we wanted to evaluate the association between FPF and various clinical markers of hemolysis in these patients. And so what we found was rather surprising, that despite controlling for disease genotype, we were able to see that FPF is significantly correlated with proBNP, which, as we know, is a marker of inflammation and often cardiac dysfunction. And we also found that FPF in the retina of these patients is positively correlated with urine microalbuminuria, which is a sign of kidney dysfunction. So to conclude, I hope that you all take away that we can look at the retina of these patients as sort of like a litmus test for an ongoing inflammation and oxidative stress that's contributing to organ dysfunction and damage that affect other organ systems in the body. We can see that FPF is significantly elevated in sickle cell patients compared to healthy controls, and that this elevation in FPF is present even prior to any structural changes to the retinal architecture. That concludes my presentation. Thank you so much.
Hi, thank you for the talk. I was just wondering, have you looked at, or has anyone looked at measuring FPF in individuals with mitochondrial disorders?
Yes.
And so like, what are the results of those and like, how do they compare to these?
Yeah, of course. There was actually a recent paper that came out, I think in just this past January at Cleveland Clinic, where the research group looked at measuring FPF in genetically inherited mitochondrial disorders. And they basically found a very similar scheme where they found that FPF is significantly elevated in those patients compared to healthy controls.
So were you able to look at neovascularization and vasoobliteration and compare that to your FPF values?
Yes, we were able to, but I haven't yet completed the analysis for that. So in order to do that, I'd have to stratify my patient population into proliferative sickle cell retinopathy and non-proliferative sickle cell retinopathy. And so we still need the ophthalmologists at New York Eye and Ear to formally classify these patients because they haven't even received their retinal exams yet.
Hi, thank you for the talk. I was wondering if there's been like a range of like normal FPF that is known. I guess so clinicians can kind of know whether they should be taking like clinical steps to prevent further degeneration and disease.
Yeah, absolutely. Thank you so much for that question. So it depends on the generation of the device that you're using to image patients. So I think with the most recent generation of the device that I've been using, the range for, I would say, a healthy macula is like maybe 20 to 33, 34. Anything above that would be kind of concerning for any levels of oxidative stress.
Thank you. Hi, can you speak to the demographic of patients and like people like controls that you used in this study? Because I do remember like seeing a flyer out for like African American controls in our class.
That was me. So I sent out me and my colleagues, we sent out an email blast to Sinai asking if people who are of African heritage could come and volunteer to serve as healthy controls for this study. And the reason why we did that was because in prior years, most of our controls are Caucasian or Asian. And so we wanted to try to race match the controls with the subjects that we have. And so now for this study, I would say over 40% of our controls are of African heritage. But we also have some Asian controls and some Hispanic controls as well.
Is there any interest in the role? I know full sickle cell disease, I'm looking at the PFP values, but is there any interest in looking at individuals with the trait? Can that manifest in retinal thinning?
So I believe that there is quite a bit of literature on, I guess, manifestations of sickle cell trait in terms of, ocular health, but I don't think we really see retinal thinning in sickle cell trait patients as much as we see it in patients who have the disease.
Okay, well, that concludes Medical Student Research Day for 2023. I really want to give another round of applause to all of our student presenters, both for the posters and for the oral. So let's, pretty fabulous. And just to really, again, thank the staff at the Medical Student Research Office, the entire medical school, Dr. Muller, Dr. Charney for making the time and the effort to come, and to thank everybody for all your hard work in putting together these amazing posters, amazing talks, and finally a last shout out to all your mentors who hopefully supported you through this whole process. So big round of applause to you all. I have two quick messages. One is the oral presenters could stay for just a minute to have photos taken. And the second is that there are still a lot of box lunches in Annenberg 5, so please go pick some up.